Well, thank you very much for coming and talking with us, Professor.
You're
Are
welcome.
you enjoying Oxford so far?
Sure. I was impressed by beautiful buildings and the green and lawn and old buildings and churches. I graduated from one of former imperial universities in Japan, and of course, a graduate of those prestigious universities in Japan are very proud of being, taught in the former imperial universities. But they are no comparison to this university.
Well, I'm glad to hear it. Well, I'll jump straight in.
Yes.
So the title of your talk, The Arms Race Between Retroviruses and Mammals.
Yes.
Who are the winners? Could you just give us a bit of background about retroviruses and what the sort of arms race is between us and them, basically?
Right, that's right. Okay, so do you know anything about retroviruses?
A little bit.
So there's a central dogma about the genetic information. So Watson and Crick said that the genetic information goes only from DNA to RNA and RNA to protein. However, there's a virus that contains only RNA as the viral genome and convert the RNA genome to double-stranded DNA, and that double-stranded DNA goes into the host cell chromosome and hide there as a part of the host cell chromosome. That's retrovirus, okay? And since the retrovirus can integrate into the host cell genome, it is a very big threat to the integrity of the host cell genome. So to survive through the repeated attack by retroviruses, we, mammals, had to develop a way to stop the infection of retroviruses. So we acquired many different ways to expel and prevent retroviral infection. But at the same time, retroviruses had to survive by infecting new hosts. So retroviruses at the same time acquired ways to overcome those host factors that restrict their replication. So there's a continuous arms race between incoming retroviruses and the hosts that try to expel retroviral infection.
Okay, brilliant. So what research inspired the title of your talk today? And
it's a long story. I have been interested in retroviruses and the host mechanisms to expel retroviruses for 30 years since I was a medical student. And when I was a medical student, there was no immunology. Immunology was newly developing. So as a medical student, I was very much interested in immune responses to viral infections. And the retroviruses are especially tricky to handle because retrovirus, as I told you, reverse transcribe the RNA genome into DNA and hide inside the chromosomal DNA of the host cells. So once retroviral DNA goes into the host cell, it's difficult to distinguish viral DNA from host cell DNA. So there's a question of whether the retrovirus is an enemy or is it a part of the host itself. So as an immunologist, I was very much interested, as a medical student, I was very much interested in how host immune system distinguish integrated retroviral genome from the host genome itself. So there must be a way to distinguish the retroviral protein from host protein, and that's a very, I thought it was a very interesting target to learn about the immune system.
So obviously I had a read of your 2012 paper as on the lead up to this, and where you focus on sort of exon 5 and the inclusion and exclusion, how that affects viral function. I was just wondering what selective pressure favors the MA3 polymorphism that results in lower
That's
antiviral
what I'm
function.
going to talk tonight. Okay,
sure.
So we were the first to find that the ApoBec3 is physiologically functioning to restrict mouse retrovirus infection. That was the first time when the ApoBec3 is physiologically actually restricting the virus in the natural host. Okay, and we found that the ApoBec3 gene in mice has polymorphisms, and some strains of mice express very high amount of ApoBec3, and the other strains express low amount of ApoBec3. And we tried to understand how those differences are made. Okay, and our first hypothesis was that the ancestors of current mice were all susceptible to retroviral infection, and by chance, by acquiring some mutation of the ApoBec3 gene, some of them became resistant to a retroviral infection and survived, and selected for more functional ApoBec3 gene. And so to prove that hypothesis, we checked many different wild mice. So there are hundreds of different species belonging to mouse genera. Okay, and we compared the genetic sequences of those different species and found that unexpectedly, the ancestral mice, ancestral mouse species, contained a highly functioning ApoBec3, and some of them all of a sudden started to express a high amount of ApoBec and acquired the mutation to reduce the amount of ApoBec expression. And we were very, very curious why they stopped producing resistant factor, And what we found was the species of mouse that is keeping the high expression genotype had integrated remnants of retroviruses that attacked them from millions of years ago. So if they are facing the continuous attack of retroviruses, they had to keep highly functioning resistant factor. But on the other hand, the species that stopped expressing high amount of ApoBec3 they do not have the remnants of incoming retroviruses, meaning that they were free of the attack made by retroviruses. So they didn't have to keep a high level expression of ApoBec3.
Okay, perfect. And so you mentioned in the same paper that over 40% of humans have at least one copy of the deletion of the genomic segment harboring the ApoBec3b gene locus. Could you theorize as to what the selective advantage of this genetic variant is?
Oh, yes. Well, one thing many people in this field know is that the ApoBec3 is not only the restriction factor against retroviruses, but it can induce mutations in host cell genome. And these days, cancer scientists have started to know that many of human cancers, including breast cancer or cervical cancer, uterine cervical cancer, contains specific type of mutations known as cataegis. And that is the cluster of mutations presumably caused by the higher expression of ApoBec3. So if you have an excess expression of ApoBec3, that ApoB3 protein can go into the cellular nuclei and can cause the mutation of the host genome. So if you are not facing the burden of retroviral infection, it might be advantageous to reduce the amount of ApoB3 expression to preserve your own genome.
Very interesting. Okay, yeah, fair enough. So what are the implications of these findings on the arms race in humans? And how can we use this research to begin to generate sort of therapies against other retroviruses such as HIV?
Yeah, it's a nice idea. I mean, a nice question. First of all, there's a big enigma about the ApoB3. ApoB3 is, of course, a DNA mutator, and it's supposed to work by inducing mutations in a retroviral genome. However, when researchers worked on the mechanisms of action of ApoB3, they actually found that ApoB3 is supposed to be a DNA mutator, but it is restricting retroviral replication without causing much hypermutation of the viral genome. That is especially true for mouse ApoB3. Mouse ApoB3 is restricting retroviral replication without causing much hypermutation. So that's called a deaminase-independent mechanism of restriction. And we have, one explanation about the mechanism is as I told you, retroviruses reverse transcribe RNA genome into DNA, and the enzyme reverse transcriptase is involved in that generation of DNA from RNA. And some people found that the ApoB3 bind to reverse transcriptase and block the production of the DNA from RNA. That's one explanation. But what we found recently is that the ApoB3 protein bind to the viral protein itself. That is the precursor of the all enzymes contained inside the virus. Okay, so this is a little bit complicated, but let me explain. The virus has its own genome and produce viral protein, but viral protein has to be cut into several fragments to compose the viral particle. Okay, so there's a viral protease that do the cutting job of the precursor protein into mature proteins, right? So what we found recently is ApoB3 is actually binding to the protease, viral protease, and hampers the function of the protease to cut the precursor protein into mature fragments, right? So what we found is ApoB3 is not just a DNA mutator, but especially mouse ApoB3 is a natural protein inhibitor, protease inhibitor. Protease inhibitor is one of mainstream of anti-HIV drug, as you know. So if you, what we are doing right now is to find out the molecular interface between the mouse ApoB3 and the mouse retroviral protease. So if you identify the molecular interface between ApoB3 and retroviral protease, then we can translate that molecular structure to a small molecule inhibitor, a new type of inhibitor. So since the ApoB3 has gone through the evolutionary selection, and it is physiologically functioning in mouse cells, the new protease inhibitor, based on the structural analysis of the binding interface between mouse ApoB3 and retroviral protease, would make a new drug that has been selected by the natural selection, therefore have not much adverse effect in the strongly functional.
Okay, brilliant. And can you theorize how we might begin to tip the advantage of this race away from the virus and into humans on a sort of long-term?
Well, it's a very difficult question. Probably you know, but the 2 million years ago, monkeys swept through the SIV infection, and the current chimpanzees are only the predecessors from the resistant family members, right? So the same thing might be occurring right now on us humans. It is known that the HIV is getting more and more resistant to drugs, more and more resistant to the immune responses. Sure. And in Japan, it is getting more pathogenic, and when I was a medical student, I mean, when I started to give my medical student the talks about the HIV, we are always saying that the incubation period to develop AIDS for HIV-infected people is more than 10 years. Okay, it's 10 years of incubation period. But now, if you get infected with HIV, about half of them had to get their therapy within 6 months. So right now we are losing the race because HIV is getting more pathogenic. But at the same time, we are developing new therapies, and of course, in the case of HIV, the prevention of infection using the good education is possible. So we are not fighting with viruses only with the biological tools, but also we can use our intellectual tools and education.
So you think a sort of the future, it's a sort of myriad of different, science as well as education and a bit of everything. That's what we need to win the battle.
Well, yes. And of course, we can use the gene therapy and the genetic editing. I don't know how much can be done for the mass of people by genetic editing, but it's, I think, applicable.
Okay.
Yeah. Well, but as I told you, in our study's case, by identifying the interface between the viral protease and ApoB3, we might be able to develop a low molecular weight medicine, and that can be used for the mass, right?
Of course. Okay. And so, yeah, we've sort of touched on this before, but so one of the most interesting discoveries from the research into retroviruses, well, at least from my point of view, is the production of retroviral vectors in gene therapy. I was just wondering what, if you could explain the science behind this, and maybe a little bit about the future of sort of retroviral vectors and their sort of importance in delivering things such as CRISPR-Cas9 tools. Well,
first of all, when the retroviruses were utilized for gene therapy, it was an almost replicable retrovirus. So it contains almost all structural genes, and so it could produce the infectious particles and could cause diseases because of the random integration of the replicating retrovirus. But after that, the new generation of retroviral vectors were produced with split structural genes and the replication of incompetent particles. And that kind of improvement of the retroviral tool would continue. And with, for example, the use of CRISPR-Cas or new targeting technology. Well, for example, we already have a new database about the distribution and expression of endogenous retroviruses. So the worries are if you use the retroviral vector as a gene therapy tool, it might be recombining with endogenous retroviruses and can unexpectedly produce infectious viruses, or can cause the transfer of the endogenous retroviral gene from one cell to another. But now we have developed and we know about the distribution and chromosomal location of endogenous retroviruses. We can manipulate the sequence of the retroviral vector so that they do not recombine with endogenous retroviruses. So that kind of an improvement will, continue, yes.
Perfect. And so on to the last question.
Yes.
What research are you sort of currently doing, and what are you most excited about in your field or in another field of biology? What are you sort of looking to and thinking, you know what, that could be the future?
Well, certainly the biologicals, antibody medicine is a very big thing. We have been using, as an immunologist, we have been using the neutralizing antibodies or anti-receptor antibodies for a long time. And we have depleted, for example, the T cells from mice, or B cells from mice, or we use the neutralizing antibodies for cytokines. And we had been thinking that those are all for experimental use. But now we are using antibodies for the therapy of rheumatoid arthritis, and we are now using the antibodies to the checkpoint molecules for the treatment of cancer. That's incredible. This is funny. In our medical school, there's a big department of the cancer therapy, and they have been working on the molecular, therapy of the cancers using new drugs or new molecules that inhibit the receptors, growth factor receptors. Something like that. And they are studying a lot about the signal transduction, and they are trying to develop new drugs that cut the signals from the receptor to the nuclei. But many of them are suddenly started working on the antibodies, and they came to our laboratory and they asked us to do a collaboration on the checkpoint blockade and the CAR T cells, So right now, immunology is, expanding a lot into the clinical field. Sure. And what we had been thinking that just for experimental use was now used actually on patients in clinical cases. That's exciting. And it will continue, I guess.
Well, thank you very much.
Thank you very much.
I'm looking forward to your talk very much.
Yes.
And I hope you continue to enjoy your stay. So
yes.
thank you very much.
Professor.